Hello students, welcome back to Nucleus Neet Coaching. I am Kiran Kumar, your biology Neet faculty. Students, today's class is going to be super interesting because we are going to discuss several interesting concepts from the chapter Cell, the Unit of Life. We are going to start with cytoskeleton, and also I am going to teach you structure of cilia, structure of flagella, structure of centrosome, basal body, nucleus and chromosome. So students, listen this class without skipping the video from beginning to end. So I will assure you that you will understand all the concepts without any confusion, but provided that you should watch the video up to the end without skipping the video. Get ready with your pen and book. Let's start the class. So cytoskeleton. What is cytoskeleton? Cytoskeleton is made up of two words. Cyto means cell, skeleton means framework. That means cell framework. Inside the cell, if you observe, there is network of proteinaceous filaments. Network of proteinaceous filaments. This network of proteinaceous filaments, it will support the entire cell from inside. So it is responsible for providing proper shape to the cell, and also it supports several cell organelles which are present in the cytoplasm. That's why this proteinaceous network, we are calling it as cytoskeleton. So generally what will come to our mind when we think about skeleton? You are right, the human skeleton. In the human skeleton you can see several bones. Okay, so the skeleton in a human body, what is its function? It gives proper shape to the human body, and also it gives mechanical support to the human body from inside. And what will happen if skeleton is absent in our body? Our body will just collapse like a mass of flesh. Am I correct? Now, NCERT definition says that cytoskeleton is an elaborate network of filamentous proteinaceous structures present in the cytoplasm. So in the cytoplasm, you can see these filamentous structures. They are elaborate. That means they are distributed throughout the cytoplasm in different directions. Okay, and they are made up of which material? They are made up of protein. That's why proteinaceous. So elaborate network of filamentous proteinaceous structures present in the cytoplasm is called cytoskeleton. Next, in which cells you can see this cytoskeleton? You can observe the cytoskeleton only in the eukaryotic cells. Cytoskeleton present only in the eukaryotic cells, absent in the prokaryotes. Got? Next, what is the function of cytoskeleton? The cytoskeleton carries out three main functions. It is involved in providing mechanical support to the cell, and it is involved in cell motility. That means movement of the cell. Next is maintenance of cell shape. It will maintain the shape of the cell. Especially in animal cells, you know students, there is no cell wall. When cell wall is absent, then it becomes very difficult for cell to maintain its shape. So in the case of animal cells, cytoskeleton plays very important role in order to maintain the shape. Understood students? Next, what are the different types of cytoskeleton filaments? Inside the cell, we can see majorly three types of cytoskeleton filaments. The first one is microfilament. Microfilament. Second filament is microtubules. Microtubules. Third type is intermediate filaments. Intermediate filaments. You can remember this by mnemonic MMI. MMI is the mnemonic to remember these three cytoskeleton filaments. M for microfilaments, another M for microtubules, I for intermediate filaments. Now let us see where these different cytoskeleton filaments are present in the cell and what is their role. First we will discuss about microfilaments. 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 So the name itself tells that they are microscopic. They are very small in size. They are proteinaceous filaments. They are proteinaceous filaments. They are made up of proteins. You can see they are parallelly arranged near the plasma membrane. So these microfilaments, they carry out different functions. What are the functions? It provides support to the cell. It provides support to the cell from inside. and also takes part in cytokinesis. What is cytokinesis, children? Cytokinesis means cell division. Cell division is called cytokinesis. So when one cell divides into two cells, at that time some constriction should be formed in the middle in order to separate the cell into two cells. So that will be carried out by microfilaments. Next is cytoplasmic streaming. Cytoplasmic streaming. What is this cytoplasmic streaming? It is a cyclic movement of cytoplasm inside the cell. As you can see here, inside the cell there will be cytoplasm. The cytoplasm will be constantly moving in a circular fashion along the inner surface of plasma membrane in this direction. The constant cyclic movement of cytoplasm along the inner surface of plasma membrane, we call it as cytoplasmic streaming. It is also called cyclosis. What is the another name of cytoplasmic streaming? Cyclosis. Okay. So what is the use of cyclosis, students? Especially in higher plants and also in protists. Cyclosis, it helps in distribution of different nutrient molecules uniformly throughout the cell. So I will show you the video of chloroplasts which are constantly moving inside the cell. Look here. Did you see, students, how the chloroplasts are moving very slowly? In all the cells you can see the chloroplasts are moving in a particular direction along the inner surface of the plasma membrane. This is what we call it as cyclosis, otherwise called cytoplasmic streaming movement. It helps the chloroplast to distribute the food material uniformly throughout the cell. Understood? Now, the next function of microfilaments is cell movement. Cell movement. So the cell movement is very important, especially in the case of white blood cells. The white blood cells, they show amoeboid movements, right? Similarly, amoeba also show movement called as amoeboid movement, where the pseudopodia are extended, and by using pseudopodia, the amoeba can move from place to place. So in order to show motility, the cytoskeleton filaments called microfilaments, they play very important role to show the movement. Example, as in the case of amoeboid movement in white blood cells and also in amoeba. Okay, that is about cell movement. Microfilaments also take part in muscle contraction. They help the muscle cells to contract. Okay, so this is about microfilaments. Next is microtubules. Microtubules are also made up of protein called tubulin. They are made up of protein called tubulin. Their function is providing support to the cell. They also help in chromosome movement, that is movement of chromosome towards the poles during the time of cell division, and also they take part in intracellular transport. What is intracellular transport? Transport of various molecules inside the cell from one place to another place. The molecules will be transported from one place to another place, okay, with the help of microtubules. That's why they are involved in intracellular transport, and they are also forming the structural component in cilia and flagella, the two locomotory cell organelles. Next, coming to intermediate filaments. Here you can see this is the microtubule, right? Next is intermediate filaments. Here you can see network of proteinaceous structures which are branched. They are connected to the plasma membrane. These black color lines, they are intermediate filaments. So what is their function? They provide mechanical strength to the cell, especially the plasma membrane and also the endoplasmic reticulum present inside the cell, and also help in cell adhesion. Cell adhesion means one cell attaches firmly to the neighboring cell. We call it as cell adhesion. This we can see especially in the case of animal cells which does not have middle lamella. One animal cell can attach to another neighboring animal cell tightly with the help of these protein filaments called intermediate filaments. Understood, students? Now copy down this. Students, now we will discuss the structure of cilia and flagella. What is cilia? Cilia are the locomotory cell organelles we can see in paramecium, and also we can see in the epithelial cells, columnar epithelial cells of respiratory tract, which will remove the particles that are accumulated in the windpipe. Okay, so these cilia are hair-like structures which emerge from the plasma membrane. They arise from the plasma membrane. Then what is flagella? Flagella are long whip-like structures. Whip-like structures. We use whip for punishing someone, right? So kings used to punish the the criminals by whipping them, whiplashing them. So whip is a long rope-like structure. If you hit it against someone, it will cause a lot of pain and injury. Okay, so the flagellum, it looks like a whip which is used to punish the criminals. Okay, so it is a long whip-like structure which will be helpful to bring locomotion in the unicellular organisms like your euglena and also in your sperm cell. Okay, so the sperm also have a long whip-like structure called as flagellum. So you should not get confused with singular and plural forms. Look here, cilia is the plural form, whereas cilium means one. Cilia is called as cilium. Flagella is plural. The singular form of flagella is flagellum. Understood? Are you clear? Now let us discuss about the structure of cilia and flagella. The foremost important point is that both cilia and flagella are seen in eukaryotic cells. They are seen in eukaryotic cells. Okay, bacteria also have flagellum, but that structure is completely different. It is not in the syllabus of NCERT or NEET examination. So we are going to discuss only the structure of eukaryotic flagellum and cilia. Cilia are only confined to eukaryotes. We don't find cilia in prokaryotes. Am I clear? Okay, look here. Both the cilia and flagella, they arise from the plasma membrane. This is the plasma membrane. Plasma membrane. In the plasma membrane, there is a centriole-like structure which is called basal body. The cilia arises from a centriole-like structure which is called as basal body. This basal body is embedded in the plasma membrane. Similarly, your flagellum. The flagellum is also arising from the plasma membrane and it is connected to a centriole-like basal body. You can see here, there is also a basal body. Okay, so according to the definition of NCERT, cilia and flagella, they are hair-like structures. They are hair-like outgrowths of cell membrane. Okay. The core is called as axoneme. What is the core? The central part of cilium and flagellum is called as core. If you cut the cilium like this, you get a cylindrical structure. Okay, in the transverse section, if you cut the cilium and flagella, you can get more or less cylindrical structure. At the center, whatever the region is there, that region we are calling it as axoneme. What is that region called, students? Axoneme. Axoneme. The central core of cilium and flagellum is called axoneme. The central core is called axoneme. Okay. Now, the axoneme has several microtubules. Several means many microtubules. They are arranged parallel to long axis. They are arranged parallel to long axis. So imagine that this is the section of cilium or flagellum. In that several tube-like structures are present. Okay, and they are calling them as microtubules. What are those tube-like structures? They are called microtubules. So these are actually filaments that provide mechanical support to the structure of cilium and flagellum, which we have just discussed in the cytoskeleton. Okay, the microtubules are elongated filaments that are arranged parallel to the long axis of cilia and flagella. Imagine that this is a concrete pillar column, a concrete pillar column. In the concrete pillar column, at the center there will be iron rods placed. So imagine that the microtubules are iron rods in the concrete pillar column. Just like the rods in a concrete pillar column, the microtubules are arranged vertically throughout the long axis of cilia and flagella. Okay. So these microtubules, which are looking like filaments, they will be running throughout the length of cilium from base to the top. So here in flagellum also, these microfilaments will be running throughout the length of the axis of flagellum. So that is about the arrangement of microtubules in axoneme. Okay. Axoneme has 9 doublets. Axoneme has 9 doublets. So in order to understand what are doublets, you have to see this diagram. So when we cut the section of cilium or the flagellum and see the section under the electron microscope, under the electron microscope you can see this type of structure. Okay, so under the electron microscope, the section of cilia and flagella reveals this type of structure. It is called as axoneme. The central core is called as axoneme, as we have discussed here, axoneme. So in the cilium flagellum section, what you can see, the structure is covered by a thin membrane called plasma membrane. Cilia and flagella are covered by a membrane called plasma membrane. This is very important. So why plasma membrane? Because they are hair-like outgrowths of cell membrane. They arise from the cell membrane. That's why they are covered by membrane called as plasma membrane. Below the plasma membrane, you can see the axoneme. What you can see, the axoneme, that is the core region. In the axoneme, you can see this rod-like, filament-like structures which are running throughout the length. So that I am representing with blue color. These are called microtubules. What they are called? Microtubules. Okay. So all these structures are microtubules. You can see here this is one microtubule. Attached to that there is one more microtubule. Here one microtubule. Attached to that one more microtubule. Two microtubules, when they combine, they form doublets. They form what? Doublets. The combination of two microtubules form doublet. So these doublets are called peripheral microtubules. In that, the doublets are called peripheral microtubules. Why they are called peripheral microtubules? Because they are present at the periphery, towards the border. That's why they are called peripheral microtubules. So one is called A microtubule, another is B microtubule. Both of them combine to form a doublet. Okay, so like that, how many doublets you can see? This is 1, 2, 3, 4th doublet, 5th doublet, 6th, 7th, 8th and 9th. Total, how many doublets are there, children? Total, there are 9 doublets in cilia and flagella. Very important. Okay, note down. So total there are 9 doublets. The axoneme has 9 doublets. Understood? How they are arranged? They are arranged in the peripheral region. They are arranged in the peripheral region. Okay. So these 9 doublets, what they are doing? They are giving support to the cilium and flagellum. Okay. At the center also, there are 2 microtubules. This is one microtubule. This is another microtubule. So these 2 microtubules are separated by some gap. They are separated by some distance. So they are called the central microtubules. They are called central microtubules. How many? They are 2 in number. Okay. So a pair of central microtubules are present in the axoneme. At the center, there are a pair of microtubules. Next, the axoneme has 9 plus 2 array. What is 9 plus 2 array? The 9 plus 2 array represents the pattern of arrangement of microtubules. 9 doublets are at the periphery. 9 doublets are at the periphery and 2 central microtubules are at the center. So what is the arrangement called? It is called 9 plus 2. 9 plus 2 array arrangement of microtubules are present in cilia and flagella. This is a very important point. Cilia and flagella shows 9 plus 2 array arrangement of microtubules. What are microtubules? Microtubules are cytoskeleton filaments that provide mechanical support. Okay. Next. Both cilia and flagella arise from centriole-like basal body. Already we have discussed there is a centriole-like basal body. Cilium is arising from centriole-like basal body and flagellum is also arising from centriole-like basal body. Okay. So now listen carefully, students. There are some connections between the doublets which I am showing in black color. Okay. These black color connections which I have drawn here, they are called linkers. They are called linkers. The linkers are the connections which connect the adjacent doublets, adjacent peripheral microtubules. So how many linkers can you expect? Can you tell? How many linkers are there? Yes, you are correct. There are 9 linkers. There are 9 linkers. Let us count. 1, 2, 3, 4, 5, 6, 7, 8, 9. Total 9 linkers are there. Very important for your NEET exam. Okay. Next. At the center, the 2 central microtubules are present in a proteinaceous covering which is called central sheath. The 2 central microtubules are enclosed in a proteinaceous covering called as central sheath, and inside the central sheath, the 2 central microtubules are connected by bridges. These bridges, they are connected by structures called as bridges. Okay. Now, the peripheral microtubules are connected to the central region. They are connected to the central sheath through structures called as radial spokes. By structures called as radial spoke. You can see here, this is a radial spoke. It is connecting the peripheral doublet with the central sheath. Here is one more radial spoke. The radial spokes are arranged in the radius. They are arranged in the form of radius. That's why they are called as radial spokes. This is one, 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 this is one. So total how many radial spokes are there? Come on, students. Yes, you are right. Total there are 9 radial spokes. Understood? So now what are these small bat-like structures, tennis racket-like structures? They are called Dynean arms. So it is not discussed in the NCERT book, but I will tell you the function of these structures which are looking like small tennis racket structures. These are called Dynean arms. They are useful for movement of cilium and flagella. Then how cilia move and how flagella move, I will explain with one wonderful experiment. Let's see. Students, I have prepared 2 ropes. Imagine that this is a cilium. Okay. And what is this? Flagellum. Okay. A cilium is a short hair-like structure that arises from centriole-like basal body, which you can see here. Imagine that this is centriole-like basal body. It is arising from centriole-like basal body. This is the cilium. Okay. Then how it moves? A cilium, it moves forward and backward like this. This is called ciliary movement. This is called ciliary movement or lashing movement. Okay. So this movement will help the paramecium to move the fluid surrounding it. It can move the fluid and it can also move the food particles. Okay. At the same time, the entire organism can also move in any direction, whatever it needs. Okay. The coordinated movements of cilia are responsible for movement of the fluid, and also it can make the organism to move in the water. So that is about cilia. Understood, students? Understood? So this is how cilia moves, to and fro. Okay. You know the boat. In the Kerala, there is a festival called Snake Boat Festival. You can see here. So in the Snake Boat Festival, there will be so many people who will be moving their oars like this. Haisa, haisa, haisa. Okay. So that paddle-like structure, it is called oar. The cilium, it works like oar in the boat. It works like an oar in the boat. It can move forward and backward, just like an oar in the boat. This is from the lines of NCERT book. Then how a flagellum in eukaryotic cell moves. How a flagellum moves in the eukaryotic cell. It may be euglena or... This is flagellum, a long whip-like structure. A long whip-like structure. Can you hear the sound of whiplash? This is called whip cracking. Okay. Look here. Here you can see there is a basal body, centriole-like basal body. This is the flagellum. Inside the flagellum you can see there will be 9 doublets of microtubules, and at the center there will be 2 microtubules. Okay. So when movement starts in the flagellum, it looks like this, like a lashing movement. Just like this. The flagellum moves like this. Okay, this is how the movement occurs in the flagellum. This is the movement. This is the movement. Okay. So by this wave-like movement, the euglena can swim in the water and our sperm can move in forward direction through the liquid. So this is about cilia and flagella. For movement of cilia and flagella, which energy is used? That is ATP energy is used. For movement of both cilia and flagella, ATP, energy currency of cell ATP is required. So then one more question you may ask, then what about the flagellum in bacteria? Flagellum in bacteria does not have microtubules. Okay, there is a special type of protein called flagellin protein in bacterial flagella, and the mechanism of working of prokaryotic flagella is completely different. Okay, if you ask me how prokaryotic flagellum will move, I will explain. Look here. This is a prokaryote bacteria. This is a flagella of bacteria. Then how a prokaryotic flagellum can move means it will move like this. Oh, you can see here. It rotates around its own axis like this. This is how the prokaryotic flagellum moves. Prokaryotic flagellum does not produce any wave-like, whip-like movement. It will rotate around itself like this. This is the movement in prokaryotic flagellum. Understood, students? Understood the concept? Copy down. Students, now we will see another cell organelle, that is a centrosome. What is a centrosome? It is a cell organelle which is found only in the animal cells. They help in organizing spindle apparatus. Let's see. Imagine this is an animal cell. This is in the state of cell division. Here towards one pole you can see there is a cell organelle which is called as centrosome. This structure is called as centrosome. Each centrosome has 2 cylindrical structures called as centrioles. So this centrosome, it will organize spindle fibers. Here also on opposite side you can see one more centrosome. This is another centrosome. It also will organize spindle fibers. So both the centrosomes will produce a spindle apparatus. So what is a spindle apparatus? 2 centrosomes along with the spindle fibers that attach to the chromosomes at kinetocores. This entire structure is called as spindle apparatus. So this is called as spindle apparatus. Okay. So this spindle fibers are made up of microtubules. Okay. When these microtubules pull the chromosomes, then what happens? The chromosomes split at the centromere region and they start moving towards the opposite poles. Each chromosome will split to form 2 chromosomes. So these newly formed chromosomes, they start migrating towards opposite poles. So this is the function of this structure called as centrosome. They help in formation of spindle apparatus, and spindle apparatus will help to separate the chromosomes towards the opposite poles, and they are found only in which cells, my dear students? They are found only in animal cells. Centrosomes are found only in animal cells. Very important point for your NEET examination. Then what is the detailed structure of the centrosome? Let us study. You can see here, this is a centrosome. Each centrosome has two cylindrical structures which are arranged perpendicular to each other. So look here, this is one centriole, this is another centriole. Both are arranged perpendicular to each other like this. Okay, two centrioles are arranged perpendicular to each other. Centrosome has two cylindrical structures called as centrioles, and two centrioles are arranged perpendicular to each other, and they are surrounded by amorphous pericentriolar material. Surrounding the centrosome there is a matter. The matter which surrounds this centrosome, it is called pericentriolar material. Pericentriolar material. So the two centrioles along with this pericentriolar material, this complete thing, it forms a unit of centrosome. Okay, and there are two such centrosomes on either sides of the animal cell. This is one centrosome, here is another centrosome. Two centrosomes will form the spindle apparatus. Am I clear? Understood, students? Now look here, let us go for the next point. Each centriole has a cartwheel like organization. Each centriole which you are looking here, this is one centriole. Each centriole is having a cartwheel like organization. You can see here, just like a cartwheel, the centriole has arrangement inside. Okay, next, centriole has 9 peripheral triplet fibrils. So when you cut the centriole in a section like this and observe under electron microscope, under the electron microscope the inner details of centriole can be seen like this. This is called the cartwheel cartwheel like organization of centriole. It resembles the cart. It resembles the cartwheel. Okay, so here you can see here, first of all, there is no plasma membrane. Plasma membrane is not present surrounding a centriole. At the center there is a protein rich region. This protein rich region is called as hub. It is called as hub. Surrounding this hub there are 9 peripheral triplets. 9 peripheral triplets, also called as fibrils. They are arranged at equal distance. They are arranged at equal distance. Each peripheral triplet, otherwise called as fibril, is made up of 1, 2, 3. It is made up of 3 microtubules. What are these rounds? These rounds represent what? They represent microtubules. 3 microtubules, A, B and C. Okay, all these 3 combined together form a fibril, otherwise called as peripheral triplet. So like that, how many triplet units are there, students? There are 9. There are 9 peripheral triplet units. Let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. Total 9 peripheral triplets are present surrounding the proteinaceous region called as hub. Now look here, one peripheral triplet is connected to adjacent peripheral triplet by a connection. This connection is called CA linker. It is called as CA linker, as it connects the C microtubule with A microtubule. That's why it is called as CA linker. Okay, next, the hub is connected to the peripheral triplets by connections called as radial spokes. You can see here, the hub is connected to each unit of peripheral triplet by a connection. It is called as radial spoke. It is called as radial spoke. How many radial spokes are there, my dear children? Can you guess? There will be 9 radial spokes. 9 radial spokes. Okay, so like this, the 9 radial spokes, 1, 2, 3, 4, 5, 6, 7, 8 and 9. So the 9 radial spokes and the CA linkers and the 9 peripheral triplets all together form the structure of a single centriole. This is the structure of a centriole. You can also see the electron microscope photograph of a centriole here. This is the electron microscope photograph of centriole. Okay. Now let us see other points. The centriole has 9 peripheral triplets called as fibrils. Central part is proteinaceous called as hub. Just now we have discussed at the center there is a proteinaceous region called as hub. Centrioles form basal body of cilia and flagella. These centrioles are not only present in the centrosomes, they form the basal body of cilia as well as flagella. Earlier we have discussed. Okay, inside the basal body also we have a cylindrical centriole which has the structure of a cartwheel like structure, which is having a structure similar to a cartwheel. Okay. Centrioles form spindle fibers in animal cells during cell division. So here we have seen that inside the centrosome there will be pair of centrioles. They will produce the spindle fibers. These spindle fibers will connect to the chromosomes at kinetochore regions and help in separation of chromosomes from each other during anaphase cell division. So the centrioles, they play 2 roles. One is they act as components of the structure called centrosome in animal cell, centrosome in animal cell, and also the centrioles also act as basal bodies in both cilia and flagella. Okay, then what is the axoneme? What is axoneme formula of centriole? The axoneme means the central core of the centriole is called as axoneme. If you observe the axoneme shows the formula 9 plus 0 array. 9 plus 0 array. The axoneme has 9 plus 0 array arrangement of microtubules. What does this 9 plus 0 represents? There are 9 peripheral triplets and 0 microtubules at the center. That is called 9 plus 0 array. Very, very important for your exam. Whereas previously we have studied about the axoneme of cilia, flagella. What is the array there? It was 9 plus 2. Here it is 9 plus 0. Okay. So this is about the structure of centrosome and the structure of centrioles. Understood, students? Copy down this. Now we will compare the differences between centriole and cilia and flagella. Okay. Students, now we will discuss the differences between the centriole, cilia and flagella. What are the differences? Look here. Here first of all plasma membrane is absent. There is no plasma membrane. But here in cilia and flagella, the axoneme is surrounded by a proper plasma membrane. This is the first difference. Next, at the center there is a protein rich region which is called as hub. But in cilia and flagella at the center there is no hub. You can find 2 microtubules. They are connected with bridges. Okay, that is the second difference. Third difference, if you observe the peripheral triplets, there are A microtubule, B microtubule and C microtubule. The 3 microtubules will form a single unit of fibril which is called peripheral triplet. But there in cilia and flagella, you can see there are only 2 microtubules which are forming a unit called as peripheral doublet. Peripheral doublet. That is the difference. Okay. So, next, the centriole is part of centrosome and it is also part of basal body. Whereas cilia and flagella, they are not part of any other cell organelles. So these are some of the differences. Next, one more important difference. The axoneme formula of centriole is 9 plus 0, that is 9 peripheral triplets are present and 0 microtubules are present at the center in the hub. Okay, whereas the axoneme formula of cilia and flagella is 9 plus 2, 9 peripheral doublets and 2 central microtubules. Students, these are the differences between centrioles, cilia and flagella. Copy down.